Imagine a world where diseases aren't just treated, but erased. A world where doctors don't just prescribe medicine, they rewrite your DNA. A world where blindness, cancer, and genetic disorders can be corrected at the source. This is no longer science fiction. This is the story of gene therapy, cell therapy, and CRISPR, the tools that give us the power to edit life itself. Welcome back to Scientific Career. This is episode 8 of our 10-episode journey through the history of the pharmaceutical and biotechnology industries. Today, we explore the rise of gene editing, the technologies that changed what medicine could become. Let's begin. For decades, scientists dreamed of fixing diseases by repairing DNA, not treating symptoms, not slowing progression, but correcting the underlying mutation. The idea was simple. The execution was not. Human DNA is incredibly complex. Every cell contains billions of base pairs. Editing even one letter without harming the rest seemed impossible. But the dream never died. And in the 1980s, the first steps toward gene therapy began. Early gene therapy was bold and risky. Scientists tried to deliver healthy genes into patients using modified viruses. The idea was to replace a faulty gene with a working copy. In 1990, a young girl named Ashanti De Silva became the first patient to receive gene therapy. She had ADA-SCID, a rare immune disorder. The treatment worked temporarily. Her immune system improved. The world celebrated. But the technology was still primitive. Delivery was inconsistent. Safety was uncertain. And in 1999, tragedy struck. A patient named Jessie Gelsinger died during a gene therapy trial. The field was shaken. Research slowed. Regulators tightened oversight. But the dream of curing genetic disease never disappeared. It simply needed better tools. In the 2000s, scientists developed safer, more precise viral vectors. AAV, or adeno-associated virus, became the star. It was gentle. It didn't cause disease. It delivered genes with high accuracy. This breakthrough revived gene therapy. Suddenly, diseases once considered incurable had hope. Inherited blindness, spinal muscular atrophy, hemophilia, metabolic disorders. The technology was finally catching up to the dream. But something even bigger was coming. A tool that would change biology forever. In 2012, two scientists, Jennifer Doudna and Emmanuel Charpentier, published a paper that shook the world. They revealed CRISPR-Cas9, a gene editing system borrowed from bacteria, a molecular pair of scissors, a tool that could cut DNA at any location with astonishing precision. For the first time in history, scientists could cut DNA, remove mutations, insert new sequences, and rewrite genetic code. Quickly, accurately, cheaply. CRISPR wasn't just a breakthrough, it was a revolution. Biology became editable. CRISPR has two main components: a guide RNA, which finds the exact DNA sequence, and Cas9, a protein that cuts the DNA at that location. Once the DNA is cut, the cell repairs itself. Scientists can use this repair process to disable a gene, correct a mutation, or insert a new sequence. It's like using find and replace on the genome. This simplicity made CRISPR explode across the scientific world. Labs everywhere adopted it. Biotech companies formed overnight. New therapies entered development at record speed. The age of programmable biology had arrived. CRISPR quickly moved from theory to real-world impact. Scientists used it to correct sickle cell mutations in patient cells, edit immune cells to attack cancer, disable genes that cause blindness, engineer crops resistant to disease, study genetic disorders with unprecedented precision. But the biggest milestone came in 2019. A patient with sickle cell disease received CRISPR-edited cells. Her symptoms disappeared. Her pain crises stopped. Her life changed. This was the first real proof that CRISPR could cure a genetic disease. And it was only the beginning. While CRISPR was rising, traditional gene therapy was also reaching maturity. Several groundbreaking treatments were approved. Luxturna for inherited blindness, Zolgensma for spinal muscular atrophy, Hemgenix for hemophilia B, Skysona for cerebral adrenoleukodystrophy. These therapies didn't just treat disease. They replaced missing genes. They restored function. They changed lives. Some were one-time treatments. Some cost millions. All represented a new era of medicine. While gene therapy targeted DNA, another technology targeted cells, CAR T therapy. Scientists took a patient's T cells, the soldiers of the immune system, and genetically engineered them to recognize cancer. Then, they infused them back into the patient. The results were astonishing. Patients with leukemia, who had no options left, suddenly went into remission. Some were cured. CAR T therapy proved that cells could be engineered like living drugs. This was biology as technology. But with great power comes great responsibility. Gene editing raises profound questions. Should we edit embryos? Should we correct genetic traits? Where is the line between therapy and enhancement? Who decides what is acceptable? In 2018, a scientist claimed to have edited human embryos using CRISPR. The world reacted with shock. Regulators tightened controls. Ethical debates intensified. The technology is powerful, but it must be used wisely. Gene editing changed everything. Drug discovery became faster. Disease models became more accurate. Therapies became more personalized. Pharmaceutical companies built entire divisions around gene therapy, cell therapy, CRISPR editing, mRNA delivery, and viral vector manufacturing. The future of medicine shifted from chemistry to biology to information. We are no longer just treating disease. We are rewriting it. The gene editing revolution teaches us that the future of medicine is programmable. DNA is code. Cells are hardware. Therapies are software. The next generation of healthcare professionals will need to understand genetics, molecular biology, bioinformatics, cell engineering, regulatory science, and ethical decision-making. This is the frontier where science, technology, and humanity meet. And you are entering the field at the perfect time. If you want to keep learning about the incredible world of medicine and build the skills to thrive in one of the fastest growing industries on the planet, make sure you like this video, leave a comment, and subscribe to Scientific Career. Your journey in science and healthcare is just beginning, and we're here to guide you every step of the way. But gene editing is only one part of the story, because once we learned to rewrite DNA, we needed new ways to deliver these therapies, new ways to manufacture them, new ways to regulate them. In the next episode, we explore the rise of mRNA technology, vaccine innovation, and the biomanufacturing revolution. Episode 9: The mRNA Era and the Future of Biomanufacturing. Do not miss it.